Good afternoon, everyone. Today we have with us Dr. Prasun Kumar, who has worked as a assistant professor at Yeungnam University, Republic of Korea, and he did his PhD from CCMB, I think. Right, sir?
Yes. No, CSIR IGIB.
Okay, IGIB, right. He was there with Dr. Shaila. Yeah. He did his PhD from IGIB, and he has long research experience, and recently also he has joined some other group as a scientist, right, sir?
Yes.
Can you explain about that, because I don't have that information. So yeah, sorry for that, but very warm welcome on behalf of Srishti. And once again, thank you so much for you have accepted this invitation in spite of your busy schedule. Thank you so much. Welcome. Over to
Yeah.
you.
Yeah. Thank you so much. To the new participant, I am Dr. Prasun Kumar. I work on microbial biotechnology and particularly applicative part of it. Recently, I started work on microbial biofilms, and I thought it would be a great topic for the students to know about biofilms and the recent advancements going on in this area. Till date, I have published more than 29 research work, and I was a assistant professor at Yeungnam University, where I was also teaching a course on bioseparation process. Besides that, I have edited 4 books, 12 chapters, and I am also guest editing for journals like Polymers and Frontiers in Bioengineering and Biotechnology. This is all about me. Now I would like to start the talk, and I chose this to keep as simple as possible, as I don't know my audience. Maybe they are graduate students who may not have the background of biology. Like in the last session that I conducted, I heard that computer science students are also attending such webinar.
Yeah.
So I will keep it simple, but the participant can interrupt me anytime if they want. All right. This topic is about biofilms, and by the end of this talk, I hope that I would transcend the message that bacteria, they behave in a multicellular fashion, and they do function like a multicellular organism. Not all the times, but sometimes. So they have the ability of multicellular behavior, and they can communicate with each other and with others. There are different types of communication, and we will know how they communicate among themselves and with others. We will also learn about the phenomena that is known as quorum sensing, and how we can manipulate that knowledge for the benefit of mankind. So the application of quorum sensing, and whether there are some benefit of quorum sensing inhibition or not. In the main screen on the left, you can see Milky Sea effect in the Arabian Sea, which is caused by Vibrio Harvey. This is a typical phenomena of bioluminescence that happens quite frequently in marine environment, but it is also quite abundant on Earth, means in soil. On the right hand side, this image, if you can see the cursor, so this image is a typical depiction of biofilm. You can see a thin transparent film of polymers on which these cells are growing and dividing. So they are rather, this is their house that I termed as a cozy house. And in recent times, people are using this term cozy house for biofilms. Okay. So
Hello, sir. Excuse me.
yeah.
Sir, please, sir, once again, sir, define, sir, biofilms, no, sir.
Don't worry. In the
Thank you.
subsequent slides, I will be explaining it again.
Okay, sir. Okay.
Yeah. I think it is not going to the next. Just a minute. I need to stop sharing and I will check.
Okay, no problem.
Yeah. Okay. So I will talk about biofilm. The whole talk is about biofilm, so I will explain it again and again. So don't worry about that. But before going into the depth, that was the introductory thing about the whole talk that I'll be discussing with you all. But before starting that, I would like to introduce you this tiny organism that we know as bacteria. A typical bacterial cell looks somewhat like this, and it consists of a capsule or the slime layer, the very outer layer of the cell. Then we have cell wall,
Hello,
and
Dr.
yes,
Prasun, the slides are not visible.
uh-huh.
I Yeah, you need to share it again, I think.
Just yeah, I forgot to do that, I guess. Yeah, is it okay now?
Yes, fine.
Okay, so I was talking about a typical bacterial cell, which has different layers and components. So the outermost layer is known as capsule or the slime layer, which covers the thick cell wall if it is gram positive and thin cell wall if it is gram negative. And the innermost layer is the cell membrane or the typical plasma membrane that every cell has. Now, besides that, we have cytoplasm, in which all the intracellular components, including the genetic materials, is present. But compared to animal cells, they do not have compartmentalized organelles. So everything available for the cells is actually present in the cytoplasm. All the machineries, all the DNA replication, protein synthesis, and all the mechanism happens inside this uh, cytoplasm. Besides that, they have genetic material, that is circular DNA, and it do not have any nucleus or defined compartment where it can keep its genetic material. So the genetic material is lying as such in the cytoplasm. Besides that, there are extra extra chromosomal DNA, that is plasmid, which provides bacteria the capability to resist antibiotics. That is the problem of current medical science, because bacteria is present everywhere, and we will see where are they present and how many they are and how frequent we interact with them. But before going into details, so we know about this and the architecture of typical bacterial cell and how they divide. They replicate their genetic material. So one genome becomes two, and similarly plasmid also replicate, and equal number of the genetic material gets transferred to the daughter cells, and it is a kind of binary fusion, right? And when we see it in a lab, we either cultivate these bacteria in flask, like we have shown here. So this is a fresh media on the extreme left, where there is no growth as of now, and after growth, it becomes somewhat milky or turbid, and by which we judge that the bacteria has grown well. Besides flask or the liquid media, we also can cultivate these bacteria on agar plate or gelatin plate, in which they grow in colonies, and the different color colonies you can see here. These small dots are termed as bacterial colonies. So this is how we work on bacterial cells, and they are ubiquitous. You can find them in soil, your gut, animal gut, anywhere, in all the living organisms. Their skin, even the blood infection, wounds. They are also present in water bodies, plants, in our food, uncooked food or the spoiled food materials, metal surfaces, even in the extreme environments. And those types of bacteria are called extremophiles. And it is very interesting to learn that even after so much of advancement in the microbiological research, we can only cultivate about 1% of the bacteria or the microbes available in the environment. So because they need stringent conditions, and we have to learn what conditions they need to grow, so we cannot cultivate all of them in the lab. So this means we still don't know much about the others that we cannot cultivate. In addition to that, human genome, for example, if you compare the inhabitants present in the human body, so number of genes present in the human genome versus the amount of bacterial cells, different types of bacteria and microbes present on our skin and within the gut, they are too much. And if you calculate, so we are like just 1 to 10% human and 90% bacteria. Why? Because the diversity in the genes uh, is mostly in the microbes, not the human cells, because human cells everywhere, whether it is your skin or it is your liver, the whole genome and the number of genes are fixed. But the number of genes present in this microbial diversity that we contain throughout our life is varying and quite diverse. And similar depiction you can see here, whether it is a reindeer or a tree, bacteria is present everywhere. And there is a problem when we talk about bacterial infection, because sometimes these bacteria, so I forgot to mention that some of the bacteria are our friends as they help in plant growth. We can use them for biofilm production, for antibiotics production, and as a nutrient supplements. So there are many uses of bacteria, and the common usage is to make curd or ferment beer, right? So we use them, and those are called friends. But there are foe as well, those that causes disease or infection, those who release toxin that are harmful to us, and those who are resistant to drug, those who spoil our food, those who make infection in or corrosion in ship hulls, in our monuments. So those types of organisms we don't need, right? And one of the trouble that we have been facing since years is about the antibiotic resistance. First, we got an antibiotic that worked very well, very nice against the pathogenic organism. But as you know that they divide and they grow very fast, bacteria grow and divide very fast. So they also changes their genetic makeup. And by those changes, they get resistance. So some of them can survive even higher dose of antibiotics. And this figure is showing the development of antibiotics throughout this past century. And you can see that they had been developing um, uh, resistance or tolerance to antibiotics, and we had been also continuously or rigorously searching for a new set of antibiotics that would be better than the previous one. So these are the versions of antibiotics that you can see on the screen. And I would like to make a disclaimer here that whatever images that I am using in my presentation, mostly they are taken from the internet online sources, and some of them is my own, so I will tell you when they will come, right? So there is no copyright issue in this. All right, so we have problem with antibiotics resistance because bacteria divide very fast and they change their genetic material. And I also told you that this antibiotic resistance property or antibiotic degradation and even production of antibiotic is sometimes governed by the plasmid they carry, the extra chromosomal DNA that they carry. And interestingly, they also share their genetic material by recombination event in the natural environment. So by having such ability, uh, they continuously evolve, and that evolution you can see in this uh, slide. So what I'm showing here is a study conducted by Harvard Medical School, and what they did, they made a big giant agar plate. So you can see that the dimension of this plate was too big. It was like a table size, tennis table, and they kept different concentration of antibiotic. You can see on the top. So first lane do not have any antibiotic, and it is increasing almost 10 times every slot. Right, and what they did when they inoculated, they saw that after 11 days of incubation, E. coli is able to tolerate as much as 1,000 times of the antibiotic. It is the same E. coli that they grew on the time zero, but after 11 days, the E. coli was able to survive even 1,000 times of the antibiotic, and this is the level of the tolerance that um, they can get in a span of just 11 days. So it is a very nice demonstration that bacteria evolves very fast. Right. So we have the problem of antibiotic resistance, and that type of resistance we call as multiple drug resistance if they are resistant to more than one type of drug, and extremely drug resistant if they are almost resistant to all the varieties of drug that we have in our arsenal uh, against this pathogen. And this problem of antibiotic resistance further increases about 1,000 times. It increases if these pathogens form biofilm. So now, what is a biofilm? It is a term coined by the Professor John William Costerton. He is also famously called as Bill Costerton. He is the founding father of biofilm research, and what he observed while he was trekking in Canada is he saw a uh, slimy layer uh, that was growing on a rock, and he thought that it is a common algae, but it is not green. Then he took that stone to his lab and checked whether it is a microalgae or something else. He found that it is just bacteria and some polymeric substances. And after that he started conducting research, and he also developed a whole research division that is devoted to biofilm research. Right. So what is a biofilm? A biofilm is a polymeric matrix that is secreted by the bacteria for their kilns and other microbes. Right. So it happens in a stage-wise fashion. So at first, so you can see the 5 stage. It is a 5-step process. Now we know a lot about biofilms. So now we know that there are 5 stages. First, in any water body or a current stream, bacteria float because they have flagella, they have motility, they have the ability to move around, swarm, swim on a media plate, or even in water. Right. So they move around, and when they find some attachment or some adhesion site, they irreversibly attach first on any surface, let it be a metal, stone, rock, anything. So they first reversibly attach to the surface and then grow on them. And once they start growing, they attach themselves irreversibly because they start secreting the substance, the extracellular polymeric substance that includes protein, uh, polysaccharides, as well as genetic material like DNA that comes out from the dying cells. Right. And since they divide quite frequently, after some time, you can see the formation of these biofilm, and these bulges are indicator of maturation of the biofilm. So in the initial phase, biofilm is just a thin layer of slime plus bacterial growth, and then as they keep on growing and dividing, their population increases to such a level that it automatically detaches. So this type of stage is called dispersion. So once it is dispersed, this type of aggregate, they again go to some other site and they attach itself. You can see that a little bit of slime is also there to help them, and occupy another space in another vicinity, right? And this cycle continues because their ability to attach to the surface remains with themselves. As they are motile, they will move around. So once they come out of the biofilm, they are ready to infect other places, right? And this is how bacteria grow and propagate throughout the natural ecosystem. Okay, so this was the overall structure of biofilm, and then I have kept 3-4 images to show you how in lab we see the biofilm. So this A, B and C here in the center is the flow cell method. Flow cells, we say that we keep liquid at one end and allow it to pass from this plate, which is quite sealed, and bacteria will grow in this enclosed section, while liquid will keep on running from one end to the other. This will try to mimic the scenario, the real life scenario of any water body. And if they form biofilm, we can check the level of biofilm using scanning electron microscope or confocal microscope, right? Then we can also check the level of biofilm formation using test tubes. So you can see if they are able to form this ring that is colored blue because of the crystal violet that is used to stain biofilms. So crystal violet stain these polysaccharides, and that's why we get these bands on the tube. And the more the bands are there, the more strong biofilm forming strain that bacteria the property of the bacteria is, right? And then we also have Congo red assay, and instead of tubes you can use 24 well plates, you can also use 96 well plates to analyze and assess the ability, how strong biofilm forming strain you have. And you can, in this case, there are some mutants also that they have tested for their biofilm forming ability. So the top image is the same, that first bacteria, all the bacteria initially they all love to remain free. They are like planktonic in nature, right? But once they find some surface to attach and the food to eat, they will attach themselves and become sessile, and the biofilm will mature eventually because the bacteria will occupy the space. They will keep on secreting this uh, extracellular polysaccharides and other materials, and these things will mature to a stage that the whole biofilm can no longer sustain an extra burden of the cells, and it starts dispersing. Interestingly, this type of phenomena is a cell density dependent manner, and that type of phenomena is termed as quorum sensing. So I will abbreviate quorum sensing as QS. So wherever in the presentation you find QS, that means it is quorum sensing, right? So it is a density dependent phenomena, and this phenomena also gives a clue to us that bacteria can not only live freely as an individual, but they can also talk with their own kind as well as to others using chemical signals, and they perform some function, right? So when there are many cells, many same type of cells, for example, if it is E. coli, if there are many E. coli around a particular area, they can perform some function. What that function could be? That function could be biofilm formation, it could be virulence factor, it could be bioluminescence, as we have seen in the case of Vibrio that do bioluminescence in marine environment. They can also initiate sporulation. At high cell density, they can also produce antibiotic, they can show symbiosis, surface motility, etc. It turns out that more than 25% of the whole genome of a bacteria can be governed by this type of quorum sensing phenomena. I will tell you how this exactly works, but just remember that more than 65% of the infectious disease is actually caused by biofilm forming bacteria. So this means the infection that we get are first they are by the pathogen, and if they can be able to form biofilm, they are further resistant, more than 1,000 times resistant to antibiotics. So biofilm aggravates this issue of antibiotic resistance. Right, and I have kept a short video just to clear whatever I said now. So I hope if it is visible to you, you just let me know. And if there is some problem, we will skip this video.
Hello, excuse me, sir. Sir, you are playing the video?
Hello?
Yes, sir. You are playing the video, sir.
Yes.
But not visible, sir.
But it's not visible to us.
Okay, then I will skip. I think it is still in the sharing form. Okay, let me try it once more.
Okay, sir.
Yeah, is it visible now?
Yes, sir. Yes, sir.
Yes, sir.
And how about the sound?
No, sir. Not fresh sound, sir.
So when I'm playing, you can hear the sound or not?
Yes, sir. Here, but not clear sound, sir.
Okay. Anyway, so I will let the video play and then I will discuss. Okay. And
Okay.
if it is not at all audible, just interrupt me immediately. Okay.
Okay, sir. No problem, sir.
Okay. Could you hear the sound or not?
No.
Okay. Anyways, so it was just a video that showed that how bacterial biofilms is develops from free cellular form or a planktonic form to a mature biofilm, and it also showed in the later slide here that amoeba or other predators eat these biofilm for energy, right? However, Legionella can grow inside the amoeba and burst the cells and get away from these biofilms. So this there are various types of microbes that can grow within a biofilm. So sometimes biofilm is formed by just one type of cells, but in a natural environment there is no control which bacteria will come inside the biofilm. So usually the type of biofilm that we observe in nature is polymicrobial in nature, right? So polymicrobial means many different types of bacteria or even yeast or extremophiles can live together within the biofilm. Okay, so I will stop this and I will go to the presentation. Right. So we learned that bacteria are able to form biofilm. And what is a biofilm? It is a slimy layer of extracellular polymeric substances that includes proteins, that includes polysaccharides, and in some cases genetic materials. Since bacteria are engaged in a or encapsulated in this matrix environment, so even if they are dying, their cell debris will remain within this uh, biofilm, right? And why it is called cozy house for the, just a minute, can you see this slide?
Yes, sir. Yes, sir.
Okay, all right. So why I said that this is a cozy house for bacteria is because just like we construct our house. Why? Because to avoid this natural phenomena and temperature fluctuations, and we want to live in an encapsulated to avoid enemies and other things. Similarly, in case of biofilms, biofilm protect bacteria from fluctuating environmental conditions, fluctuating pH, salt conditions, less food, more food in situation, and then they also occupy the space. Suppose they find some food and they start growing on them and they develop biofilm. So now they occupied the whole space. So it is good for them that they have a food source now, at least till the time they can utilize that much of food, and once it exhausted they will leave their biofilm lifestyle and infect other places, right? So it is a favorable environment where bacteria want to relax, it don't want to struggle more. In a free form it has to move around and try to find food wherever it can, uh, uh, it can find. Suppose for example dead animal, nearby dead animal, the proteins, the carbohydrates will be available in plenty. But once it goes, or suppose washed away by the water current, then it has to move again some other places and divide and grow and divide like that. So that's why this type of structure is a cozy house for bacterial cells, and it happens, it gets constructed by the phenomena called quorum sensing. It is a density dependent phenomena. This means until they reach to a particular cell density, they will not form biofilm. Once their cell density reaches a threshold, then only it will start expressing genes that will perform the functions such as listed here, like biofilm formation, virulence, bioluminescence, etc. So bioluminescence is quite unique in the aspect that uh, we can measure it, we can measure bioluminescence. Okay, so that's why people use Vibrio harvey or Vibrio fisheri strains to score or to regulate their assay system. And that's why for quorum sensing studies, bioluminescence bacteria that can perform bioluminescence is good model organism. Okay, while in other cases where people want to just work on pathogens that are forming biofilm, they can use crystal violet stain for rapid screening of various anti-biofilm agents. So we will talk about it later. Before that, I would like to summarize the things. Uh, Bacterial cells are commonly considered as asocial, means they live freely, and they are abundant in human body, right? Besides that, they also perform very important function in human lifestyle. For example, in fermentation, in wastewater treatment, we use them for metal recovery, but they also cause some issues like ship corrosion, metal reduction and corrosion, all those things. Right, and how people got to know about this type of phenomena that we termed as quorum sensing. So it all started with a squid, and the work was done by Professor John Woodland, also nicknamed Woody Hastings, in 1970s. So it is very new field of research. So what he observed is that squid host bacterial cells, that is Vibrio fischeri, and what it does. So this is the squid he worked on. It is a Hawaiian squid, lives in the Hawaii area island, and it has light organs that host or that contains Vibrio fischeri, a lot of Vibrio fischeri within this light organ, and how it uses this Vibrio fischeri and the phenomena of quorum sensing for its survival. So there is two aspects. First aspect is for the squid itself, for the survival strategy. And what is the strategy? Particularly when there are a lot of stars and moons, squid can use this bioluminescence as a torch, and if there is light on the top, so squid will have sensors that will detect the amount of light that is coming to its body on the top, and then equal amount of light it will try to open and shut its light organ, so that equal amount of light will also go at the bottom. So by doing this, what essentially it is uh, trying to do is, it is trying to avoid making shadow, or it is avoiding the predators, because predators can see its shadow and will identify the squid and it will catch it. But if it is releasing some light, it will confuse the predator, and it is a kind of camouflage strategy that squid uh, adopt to avoid this predation. So for squid, there is a gain by keeping Vibrio fischeri, but there is a catch also. What Woody tried to answer is how this organism actually is regulating this type of bioluminescence, how it is controlling the amount of light. So what he understood later on, that in the daytime when squid is hiding, because it is a nocturnal animal, so it only comes out for hunting and food in the night, while in daytime it hides in the sea bottom, in the bottom of the sea, right? So during that time it squeeze out extra bacterial load that it has in its light organ, so it is diluting the amount of bacteria that it has in its body. And by the time night comes, the bacterial cells will divide again and will reach to the cell density, so that it starts glowing, and it will use this type of phenomena in a cyclic manner to avoid predation. So this was the groundbreaking research, and it has evolved to a very advanced stage now. And now this quorum sensing phenomena has been related to biofilm and uh, uh, all other biochemical aspects that I have listed here, like virulence factors, sporulation, antibiotic formation, symbiosis, etc. And this is how bioluminescence look in a laboratory. So this is an agar plate where you can see the biofilm forming bacteria, bioluminescence bacteria, and this is the illuminating flask containing bacterial culture. Okay, so Woody Hastings found this phenomena and termed this as quorum sensing. But then Mike Silverman, he did the genetic work to understand how exactly quorum sensing happens within the bacterial cells, and he coined the term for the gene that governs quorum sensing as lux. Lux because it is the god of light, in their system, in their religion. And then S is for synthase. So Lux S means it is the synthase that is responsible for the synthesis of chemical that ultimately leads to bioluminescence, right? And there are many signals that we will talk about now. So these chemicals that they release when they are growing in any system, for example, AHLs, the first and the foremost signal that was found in Vibrio, Serratia, Ralstonia, and many other organisms. So there are many quorum sensing signals, and why they are called signals, because these are the molecules that are identified back by the bacteria and then they perform the function. So there are more than 17 types of quorum sensing signals. So these are the family of signal. For example, once you say AHL, this is acyl homoserine lactone, means it will have a acyl chain and then a lactone ring, right? So all the family of the signal of this class will have a lactone ring that is fixed, but their carbon chain length will vary. So say for example, C4 means there is 4 carbon chain, acyl chain, and then there is 6 acyl chain, 8, 10, and there are oxo form, hydroxyl form also. So there are many variants in this class, and different organisms produces different types of signals. Besides that, so AHL is mostly found in gram-negative bacteria, but gram-positive do have their own quorum sensing signals, and they are mostly peptides, very short peptides. Besides that, recently people have also identified indoles, AI2, that is autoinducer 2, and then diffusible signal factors, butyrolactones, quinones, pyrones, farnesol, etc. So there are a variety of quorum sensing signals that have been identified slowly after 1970s. So how it works? So when bacteria is at very low cell density, so AHL synthase, that Lux S, for example, in case of Vibrio, so bacteria will start synthesizing these chemicals that is called quorum sensing signals. These signals will keep on releasing from the bacterial cells. At low cell density, outside of the cell will have quite few amount of signal, but once they are grown a lot, means the bacterial cells have divided and their population is too much, that means they are in very high cell density, they are very high in number, the number of molecules, these quorum sensing signals outside the cell will be higher than the intracellular uh, cytoplasm, right? And that's why these signals will diffuse or it will be received by the transmembrane receptors, and it will regulate the gene expression by their cognate receptor. It is similar to the lock and key hypothesis or the enzyme substrate complex. So you can consider this receptor as the enzyme and this molecule as their substrate. Right. So once they receive these signals, there is phosphorylation and dephosphorylation of the downstream proteins happens. Ultimately, it leads to activation of the target genes. And the target genes can regulate either biofilm formation, virulence factor, or sporulation, etc. Professor Bonnie Bessler is another pioneer scientist who has worked on Vibrio, and some of the contents in this slide has been actually taken from her work. So this type of AHL signaling is very common in gram-negative bacteria, while gram-positive bacteria, they releases oligopeptides, and since they are peptides, they cannot diffuse easily in the cellular compartment, right? So they should have some receptor proteins that will transfer the signal from outside to inside the cell. Similar to bacteria, fungus also have signaling system, and the common fungus that is studied for biofilm formation is Candida, and the property that people look after in Candida is their hypha formation as hypha means virulence. So hypha, if they are able to form hypha, means they are ready to infect the host. So if we can stop hypha formation, this means we can stop this quorum sensing uh, whole setup of quorum sensing system within the fungus, right? So similar to bacteria, the fungus will also produce these signals by signal producing proteins. This triangular shape is the signal quorum sensing molecules, and then these molecules will be received by the signal receptor proteins, and it will transfer these signals to the intracellular proteins that will ultimately activate the proteins binding to the promoter, and it will lead to the expression of genes that will govern virulence, growth, and their morphogenesis. And the hypha formation actually comes in morphogenesis part of the fungal cell. And as in bacteria, we have seen there are many different quorum sensing molecules. Fungus also have different signal molecules. And here is the list of the molecules that have been identified to be involved in quorum sensing in fungus. So you can see the Candida albicans, Zygomycetes, and Euromyces have unique compounds that they utilize for quorum sensing phenomena. And these are the structure of the signals. You can see the structures varies a lot. Okay, and this hypha formation I was talking about. So once you have quorum sensing molecules, and the fungus or the yeast will start forming either they will just form bud or they will form long mycelia. So it depends on the amount of the quorum sensing molecule that is present within the environment, and bacteria will change its morphology. That's why quorum sensing is involved in the morphogenesis of fungus. So by now you may have noticed that from just quorum sensing, I started talking about many different compounds that are used as quorum sensing signals. And this indicates that bacteria are having their own language, and they can be using just one chemical language, or they can use many different chemicals for communicating among themselves, right? So bacteria or fungus can be multilingual, right? And if they are using this for their personal communication, means just among, for example, if it is Vibrio fischeri and it can only communicate with another Vibrio fischeri only, then we call it as intra-species signal. Okay, so if it is just Vibrio talking to Vibrio, but if Vibrio talking to E. coli, if it is Vibrio talking to Pseudomonas, and vice versa, we call it as a trade communication or commercial language, and that type of phenomena is called interspecies communication. Besides these two types, there could be inter-kingdom communication as well, and we will talk about such molecule later on. So by now, we would have a clear idea that we must study bacterial language. And why? Because bacteria are highly adaptive to fluctuating environments, and they are also getting resistance, antibiotic resistance. And if we can stop this type of communication that they do through quorum sensing system, we can at least we can stop them from getting into a cover of biofilm. And because of these biofilms, they get about 1,000 times resistant to antibiotic treatment. So that's why studying such phenomena is important. And I already talked about this, that more than 65% of the nosocomial infection, particularly the infection caused in this hospital environment, are caused by the biofilm forming pathogens. There are many practical implications of such system. If we are able to stop, for example, Pseudomonas infection, So it would be a great for the cystic fibrosis patient, for immunocompromised patient who are getting infected almost every day by Pseudomonas. Pseudomonas aeruginosa and Acinetobacter baumannii are the two opportunistic pathogens that are quite deadly in real life. So once we are saying that if we can stop quorum sensing, what we are essentially saying is we want not to kill the bacteria, but just making them either mute or deaf. What we mean by that is uh, muting them so that they cannot talk, or stopping them to receive the signal so that they become deaf and they cannot listen even if they are more in number. So by doing any one of them or both of them, what we are doing is we are stopping their communication system, and so that they cannot form biofilm. And the I think okay. So how we can do that? I am taking a common signal, quorum sensing signal, that is acyl homoserine lactone. So this is the structure of AHL, and where we can degrade this signal, we can degrade at point 1, 2, and 3. So we can use different enzymes. There are two already known enzymes, that are lactonases and acylases, that degrade at these different points. So acylases will degrade the acyl chain, while lactonases will degrade this lactone ring. Right. So by degrading the signal, you are stopping them from communicating. Right. So we can stop the signal. We can stop them by from receiving the signals, that I will come later on. In here, you can see that if we degrade the signal outside the cell, bacteria will keep on trying to communicate, but we are stopping them from communicating. Once the signal goes out, we are degrading it using enzymes such as lactonase or acylase. Right. Another strategy could be we can just stop them here itself in the intracellular environment by using some inhibitors that stop this signal synthase to produce the signal. So if this enzyme will not work, bacteria will no longer produce the signal. Right. So this is a kind of making them mute. While in the other case, you can you can use the inhibitors that will have a similar chemical structure as that of the signal molecule, and so there will be a competitive binding with the receptor, as happens with enzyme systems. So due to the competitive inhibition, the signals may no longer bind to its cognate receptor, and there will be no signal relay and no biofilm at all. So there could be different strategies to stop this type of bacterial communication. Similar strategy can also work on fungus. And there are some quorum sensing inhibitors from the fungus, particularly from Penicillium and Delcia pulchra. And furanone is quite well-known compound, as it competes with the lactone group of the AHL. So you can see the structure is quite resembling with the lactone ring, but they are having other R group, and it varies with different species. And it was identified by Rasmussen, quite highly cited paper he has in this regard. So there could be various approaches of quorum sensing inhibition. The one that I talked about already, using biological system or enzymes. But in addition to that, we can use substrate analogs, we can use natural furanones, we can use alkaline or pH conditions to degrade the signals, and we can also use another natural uh, blockers such as diketopiperazine that will hamper their communication. So it depends on the target organism which we are targeting and the type of signal that they are secreting. And ultimately, whatever we do, ultimately we have to see such effect of inhibiting quorum sensing system in a live model. And the commonest model that people use is C. elegans or a mice model, where they infect the C. elegans or mice with the pathogen. Then they treat the infected organism with quorum sensing inhibitors, and they try to see whether they died or survived. If the death rate decreases, this means the quorum sensing inhibitors actually help the model organism to survive, right? So, for example, we have a Pseudomonas strain that is highly pathogenic, and we infected the mice with Pseudomonas that is also able to form biofilm, and then we added quorum sensing inhibitors. So what it will do, it will stop the bacteria to communicate, it will stop the bacteria to form biofilm within the mice, and so they are unable to form biofilm. But Pseudomonas is still dividing, right? So Pseudomonas infection will be there within the infected mice, but the virulence of Pseudomonas will not occur because it cannot communicate, right? And since it is unable to communicate, essentially what will happen is the innate and adaptive immune system of the mice itself will clear the free living pathogens, that is Pseudomonas, and ultimately the mice will live or survive. So this type of system people have speculated in the early 2000, and they have also demonstrated good quorum sensing inhibitors that helps them to come up against the biofilm forming pathogens. Now, besides quorum sensing inhibition, there are another aspect of biofilm, and that is the dispersion, biofilm dispersion. And what it is it is a natural system of where bacteria can identify the amount of load that is going on in the mature biofilm. So once the biofilm is getting towards maturation, they know the load, and they start signaling another type of signal, so that all the bacteria will try to leave the system. Okay, so and this phenomena is called biofilm dispersion. They are dispersing from their mature biofilm, right? And one such signal molecule that recently got identified is called diffusible signal factor, or DSF. They have a characteristic cis2 double bonds in them, and it is a fatty acids. So what it does, once the bacteria receive this type of signal, again phosphorylation and dephosphorylation happens in this receptive end of the transmembrane domain of the bacteria. Here I am showing a Burkholderia cepacia signaling system, where you can see that the cyclic digmp levels is going down. By degrading this cyclic digmp, the bacteria is going from a biofilm forming stage to planktonic cells. And what type of genes that they are activating? They are activating mannase and I think xylanase or xylose degrading uh, enzymes, so that they can degrade the polymeric substance of the biofilm and they can live freely outside. So this type of signal is called biofilm dispersion, and DSF is one such signal. And instead of making some quorum sensing inhibitors, we can also utilize this system, so that we can induce bacteria to leave the biofilm life cycle and go towards planktonic cells. So the question arises, can we use fatty acids? Fatty acids are already known as a broad spectrum antibiotic or antibacterial agent. But interestingly, at higher concentration only it works like that, but at lower doses, it only acts as an antibiofilm agent. So at higher concentration, you can see there are many different method to for its antibacterial mechanism. For example, first it can start leakage of the cell membrane. As they are fatty acid, they will get integrated within the cell membrane, and it will lead to cell lysis. Besides that, fatty acids will disrupt the membrane potential so that the proton motive force will no longer be active, and and this will subsequently lead to hamper of ATP synthase. So less ATP synthesis will stop the bacterial growth and ultimately it will die out. Right. Besides that, it also fatty acids also inhibits nutrient uptake and also inhibits fatty acid enzymes involved in fatty acid biosynthesis as well as metabolism. Right. There are many fatty acids like saturated and unsaturated fatty acids that had been studied, and I have recently reviewed all those fatty acids for this property of antibiofilm at lower doses. So in different organisms, they behave or they act differently, but there are still some questions that are yet to be answered. For example, how FADR is talking to TOX-T, means how FADR is activating TOX-T, we don't know. And there are many other things, how fatty acids uh, affect non-QS targets, because QS targets are known, for example, in Acinetobacter baumannii, but not in other cases. So there are many aspects of biofilms that is coming in recent times, and fatty acids are one of the attractive target. Why? Because it is natural and abundant in our ecosystem. Particularly in plant cells, we have many different varieties of fatty acids with different R group. And another group of researchers have recently found, by the proteomics study, they have found out all the possible targets of fatty acids in Candida in fungus. So this work was on mostly on bacteria and Candida also, but they have only worked on Candida and they found many different targets, and that targets include ergosterol synthesis, hyphal formation, biofilm formation, sphingolipid, and oxidative stress. So all these red arrow, the down arrow colored red, they are getting repressed by the action of these fatty acid. So the myristic acid is a C14 fatty acid. They used very low concentration of just 125 microgram per ml, and they got this much of inhibition when they did proteomic analysis. So, prevention of biofilm formation or induction of biofilm dispersion is of utmost importance in the battle against infectious disease. And there are wide range of distribution of fatty acids. As I already mentioned, that in nature we have many varieties of fatty acid, and it starts from C4, from volatile fatty acid, to waxes, that is C38 and even C56. So there are a variety of fatty acids present in the environment, and plant uses such fatty acids for to fight against these natural pathogens. So it is a kind of their innate defense system, right? So this was all about biofilm and quorum sensing, and they both are linked together. So I could not avoid going towards or discussing more about quorum sensing system and how we can use it to avoid biofilm formation. So I hope this would be clear to you. And then this was all very good, very how to say, hopeful things that I talked, but there is a catch, and that we should always be aware of. We should not become overconfident of our inventions, as once we found antibiotic and we thought that we won the match, but that was not the case. Bacteria evolve very fast, and such thing also happens with uh, these quorum sensing inhibitors. What recently people have pointed out is that bacteria can generate resistance to these quorum sensing inhibitors as well. And how they can do it? They can use their multiple efflux pumps. It is the same similar pump that they use to pump out excess of antibiotics or other toxic materials. So if they use multiple efflux pump, or if they are not using just one signaling system, if they have multiple signaling system, and then we have to block each and every type of communication system that they have. As I said, that they are multilingual. Bacteria are multilingual, so we have to stop all type of communication to avoid the biofilm formation. But then that becomes tricky. But nevertheless, the amount or the number of quorum sensing inhibitors that we have identified till date, and we are still developing just in parallel to antibiotics, we are also developing various derivatives of quorum sensing inhibitors, and possibly by using combinatorial drug treatments, this means antibiotics plus quorum sensing inhibitors, we may try to avoid the problem of resistance. So this was all about biofilm, its application, as well as how we can utilize the knowledge of quorum sensing and stop bacterial communication to against the pathogenic organism. So if you people are interested more, I will go ahead with another interesting molecule. Otherwise, I would like to stop here. Hello. Hello.
Yes, sir. Hello.
Yeah, shall I go ahead, or
Yes, sir.
is there any question in between I would like to ask? If there is some question, I can address it, because again the track will change a little bit, so I am stopping it here to address some question, whatever it is.
So there is question in chat box.
Okay, yeah. Is quorum sensing signals are the primary factors which helps in both formation and disruption? No, quorum sensing signal is the DSF, or the biofilm dispersion, is a type of quorum sensing, but it is not like conventional quorum sensing phenomena that we discussed for biofilm formation. Whether the composition of biofilms of different microbes are same or different. The composition of biofilm changes even with the same organism. The type of EPS, the extracellular polymeric substances, also changes. The amount of protein they release, the amount of enzymes present within the biofilm, these all the things changes with the environment and with the, for example, if we are working in lab, so we are giving them some liquid media to grow, and if we change the media composition, or if the bacterial cell division is at different stages, the amount of enzymes that they will release within the biofilm will differ. And when you will do the FTIR analysis, you will see that the amount of protein characteristic band corresponding to the protein or DNA will differ. The death rate within the biofilm also vary with the maturation phase and the growth phase of the biofilm, right? So all these factors affect their composition. So the biofilm composition will always vary. If it is just a single type of cells, the composition will be different. And if it is a polymicrobial biofilm, then you can anticipate that each type of bacteria, each different species will secrete different enzymes, and all those enzymes will be present on the matrix, extracellular part of the cell, right? So I hope I have addressed both these questions. So if you are with me and you want to learn more, because this is just my work that I will be discussing, so I will continue. So can you see the slide?
Yes, sir.
Yes, sir. It's visible, sir.
Yes, sir.
Okay, so till now I talked only about intra-species signals and interspecies signals that that bacteria use for quorum sensing and biofilm formation. Okay, so also remember that once we are saying that it is a quorum sensing, it may be virulence, not biofilm only. But once we are saying only biofilm, this means it is only about biofilm, right? So besides interspecies and intraspecies communication, some signals are also involved in interkingdom signal. For example, indoles are one such signal that was recently promoted. It is a kind of archetypal hormone. This means that it is present in all forms of life, but it started since the beginning, like when the first unicellular organism came into picture. The indole synthesis starts from the degradation of tryptophan. So the bacteria uses tryptophanase enzyme to degrade tryptophan into indole, pyruvic acid, and ammonia. So only these three products will be formed by the degradation of tryptophan. So one will be the indole ring. Okay, and you can see that ring in this dietary tryptophan chemical structure here on the right side. So in this hexagonal structure with this 5 carbon compound, this all together is called indole with the NH group at the bottom here. Okay, so once bacteria degrades tryptophan, it releases indoles, and indole is involved in many different interkingdom signals. In this slide, I am showing how it affects our health, means animal health or a mammal health, by communicating differently through gut organ axis. What is that gut organ axis? We know that many microbial pathogens as well as other non-pathogenic species uh, who inhabit our gut or the colon, and whatever we eat, including protein and sugar molecule, they also degrade it. Okay, so some beneficial organisms such as probiotic organism, that is such as bacillus, lactobacillus strains, bifidobacteria, so they are probiotic that help us degrade the food, but some also are pathogens. Okay, so dietary pathogen, dietary tryptophan. So the degradative part of the protein releases these tryptophan as well as other amino acids. But E. coli and other pathogens convert these tryptophan into indoles, and indoles can be just indole group or iso or indole propionic acid, indole aldehyde, indole acetic acid, or indole lactic acid. So there are a variety of indole compounds that will be released within the gut. And what will happen if they are being released in the system? It can be beneficial for our gut, or sometimes it can be harmful. How it will be beneficial? So first, these types of indole compounds, it will stop the inflammation or the colitis within the gut. So colitis happens when there is some infection within the gut, and this type of inflammation or infection is stopped if there are a lot of indoles present in the gut. Right. So the bacteria that is releasing indoles are good for our health, our gut health. Right. They also activates our gut immune cells to clarify or to clear out these types of infection, as well as to reduce the inflammation by secreting cytokines. And secretion of cytokines such as IL-22 or IL-10 also helps to stop further infection. For example, IL-10 will induce the secretion of mucin. So there will be a lot of mucus layer in the gut, and since there are more mucus in the gut, the chances of infecting these epithelial cells will be very less. Right. So by increasing the mucin content within the gut, we are helping ourselves to reduce the infection, as well as this mucin also helps the passage of food by the peristaltic movement, right, during the digestion process. So indole helps in both the cases, like digestion, inflammation, gut health. Overall, it is beneficial for us. And how it is coming out? It is coming out from the dietary tryptophan, and the gut bacteria is producing these indole compounds, right? In addition to this, IL-22 also induces antimicrobial peptide secretion by the epithelial cells. So in the presence of IL-22, how IL-22 came into picture is by the activation of ILC3, which is activated by the indole compounds. So once indole is in the gut, it will activate ILC3 to release IL-22, and after reception of IL-22, epithelial cells will secrete antimicrobial peptides. And since the name itself suggested that it is an antimicrobial peptide, so it is a peptide that is antimicrobial in nature, right? So they will stop the growth of pathogenic bacteria, right? Besides that, they can induce L cells. L cells, what they do, they secrete GLP-1, and GLP-1 reduces our appetite. So if we are eating more and more protein containing tryptophan, we will reduce our hunger, because if our L cells will get indoles signal to stop um, the feel that we are hungry, so they will stop or reduce our appetite, right? Besides that, the gut permeability and gut homeostasis, so all these things are actually coming under gut homeostasis. And gut permeability is the tight junctions. So so the indoles also regulate the mucin production as well as tight junctions, so that if the tight junctions are not proper, infectious pathogens can trespass from the gut to internal organs. So tight junctions have a very important role to play in our common health. So this, till here it was gut, this blue dotted line. Till here we were talking about gut environment and the indoles that is present in the gut. But ultimately, indoles get absorbed through the blood circulation and it goes towards different organs, and that's where the term that is called gut organ axis come into picture. So if it is brain, we will call it as gut brain axis, gut liver axis, gut kidney axis, like that. So indoles from the gut will reach to liver, and they will be converted to indoxyl 3 sulfate or indole 3 acetic acid, and these two are toxic to our health. They are uh, uh, they cause renal toxicity, they blocks our renal tubules, and that's why they are considered as toxic materials. And if we have very high quantity of indoles, or at least if we get very high quantity of indoxyl 3 sulfate in our liver or in the blood circulation, it will become difficult for our kidney to clarify them, right? But if we have indole propionic acid, so if we have IPA, more and more IPA in our blood circulation, it stops renal toxicity. This red bar means stop. Although the exact phenomena is not known, how it is actually stopping this renal toxicity caused by indoxyl 3 sulfate or indole acetic acid. But this is an interesting thing to look out for. Besides that, IPA or ILA, IPA is indole propionic acid and ILA is indole lactic acid. These both are helpful for neurite growth and its activity. So even our cognition behavior gets improved if we have indoles. Some of the indoles are also helpful for hormonal balance that we have. So for example, 5-HT, that is 5-hydroxytryptamine, it is serotonin that governs our mood and other things, melatonin that is also involved in circadian rhythm, and isetine. These three things also helps in Alzheimer disease, in our emotion, and also works as a neuroprotectant. Okay, so indoles are involved in these many things, but now you will be surprised when I will say that human body cannot synthesize indole, right? So this means we have to rely on bacteria to get all these indoles, and indoles are important for our gut health as well as for the hormone synthesis, for example 5-HT or melatonin, right? So gut bacteria is equally important for our health, and indole represents an interkingdom signal in this case. So here I have mentioned that some of the indoles, there are enzymes like CYP2E1 and CYP2A that is present in the liver. Okay, and the indole compounds that comes from the gut, whether it is indole or iscatol, its derivative, or the indolacetic acid itself. So they will all go towards liver and gets converted into a variety of compounds. Some will be clarified by the kidney, but others will go and do their respective job. And the majority, the main or the unique thing that they do is they produce 5-HT, that is serotonin. It governs cognition, memory, nervous system, bone metabolism, and organ development. Besides that, we have melatonin that governs circadian rhythm, means it impact our sleep behavior, antioxidant, and immune system. Among these indoles, indole propionic acid is an important antioxidant, and indole is very commonly found particularly in cruciferous vegetables. So if we are having broccoli or other cruciferous vegetable, we will be having plenty of indole in our gut as well as in our body. In plants, indole helps to prime the plant against the infectious these insects. So, for example, aphids infect plants, and these caterpillars also infect plant and damage their leaves. But once this damage starts happening, the enzymes here will start releasing indoles, and the more and more indole production will happen. It will go towards the nearby plant and it will prime it. Prime it means it will start the defense signal of the plant. So this is a representation of maize. So if maize plant if is infected by a caterpillar, so the degrading leaves will send signal to the adjacent plant so that they will start activating their defense signal, and further infection of this type of caterpillar will be avoided by their activation of what is that plant defense system that is always innate in nature, like gibberellin acid will be more in this adjacent plant, right? Besides that, the indole that is secreted due to this damage, they will repel another herbivores, for example these organisms. So it will give them signal that it is already being damaged by others, so it is already a spoiled site, so there is no need to go. They will find some other leaf to infect on or to eat on, right? Besides that, indole, if it is present in a flower, so it will attract pollinators like bees and even flies, right? So in case of a carrion, a dead animal, they have protein, and bacteria infect these uh, dead animal body and they degrade it, and during this spoilage indole get released, and this is the reason a lot of flies goes towards that dead body, and they think that it is a good oviposition site as there are plenty of food available for their offspring, right? So indole acts as a signal for the flies to for oviposition as well as mating site. So indole is an attractant for honeybees, pollinators, as well as common bee. In addition to this, indole also attract beetles that that attack the parasitoids. So what is happening here? Indole is working as at a multiple level for plant life cycle. First, it is initiating their defense response and helping them to prime themselves. They're acting as a repeller of parasitoids which infect their leaves. They also recruit or help in the recruitment of these predators, that is beetle, which actually kill these parasitoids, right? And besides that, they also attract pollinators for the plant, and in other cases, for example, flies. For flies, they act as a signal for oviposition and mating. So there are many roles that indole play, and all it is doing is through the degradation of tryptophan. And the tryptophanase gene is exclusively found in bacteria, not in plants, not in animals. And this is a very interesting paper of 2020 that also said that besides these animal and plant, indole also have a role in planaria. Planaria, you may have known that it is a unique organism. Uh, from wherever you cut this organism, it will again grow to its full shape. So it is studied for its regeneration capability. And what scientists have found out that there is a common symbiont in this planaria that help them secrete indoles. So the organism is, I think, Aquitelia. So these Aquitelia will secrete indoles, and it confuses the planaria by inhibiting the WNT signaling. So naturally, if you cut from this side, so if you cut from the half, the head portion will develop a tail and tail portion will develop a head. But in case of this, when there is indole present in the system, it will stop the WNT signaling and it will confuse it. So instead of developing a tail, the organism develop another head. So two-headed planaria can be observed by the treatment of indole. So this type of phenomena indicates how bacteria is, or the bacterial signals are important in all other life forms. In some cases it is beneficial, but in other cases it is harmful. So recently I did an in silico analysis because we already saw that these are the varieties of organisms that indole affect. Okay, indole affect the marine organism. This is sea cucumber, sea anemone, planaria, C. elegans, even flies and mosquitoes, even culex gets attracted for their oviposition and feed location, and plant priming, as well as ourselves, that is human. So what I did is I tried to find out from in how many organisms do we have tryptophanase, and is there any link up with any higher organism, means whether some higher organism have this tryptophanase or not. So I digged out the NCBI database for the presence of tryptophanase enzyme. Before discussing that, I would like to tell you the basic thing that the life form is divided in this hierarchy. So we have a big domain in which contains a lot of kingdoms, and then we have phylums, then various classes, and then we have orders, family, and then genus. So very commonly we talk about just genus and species of bacteria, but they also belong to a particular family, to a particular order and class, and so on. Right. For example, if you are talking about Escherichia E. coli, so we are talking about a genus Escherichia, and species is coli, and it is a gamma proteobacteria. So this is their family, order, and class is in this manner. We will write in this manner. Okay. So after analyzing the NCBI database, we got to know that there are about 95 genus that have TNA gene present in them, and you can see that among these 95 genus, there are many of them are gram-negative bacteria here in this list, and some of them are gram-positive listed here, maybe 8 or 8 organisms are gram-positive bacteria. There are, interestingly, there are many archaea, some fungus, and some eukaryotes. So I told you that not all higher organism produces indole, not even plant, but you can see that small organism like fungus produces indole by tryptophanase enzyme. And we also got hits in Drosophila, that itself is an insect, and horseshoe crab and all others. But when we saw it in deep, we found out that it is close to tryptophanase. So it is tryptophanase like, but they do not have a very close similarity to E. coli tryptophanase. So we ruled this out when we are talking about higher organism. So we focused only on the unicellular organism, means unicellular prokaryote as well as eukaryotes. So this was the distribution of the gene. So we have 50 genus of bacteria, 19 archaea, 7 fungus and 20 other eukaryotes. And we used some parameters to delineate which gene sequence we will use for the analysis. So out of the thousands of gene sequences available, we narrowed it down to just 221 sequences, and above is the distribution of those sequence. We used MegaX software to for the phylogenetic analysis of all these uh, tryptophanase gene sequence. And this is the gist of the whole study, where you can see that the tryptophanase gene is quite conserved in all archaea. All were halophiles, mostly. Most of them were halophiles, and only one thermophile was found in this study. Besides that, these light blue color are the gram-negative bacteria, and they are also well organized at a corner. So this means that they are also quite conserved, and these color are mostly for uh, gamma proteobacteria. And you can see that some of the protists are unicellular eukaryotes. So eukaryotes are coming in a very different group, and some of the gram positive bacteria, that is green in color, represented in green, are also adjoining them. So this means that the origin of tryptophanase in protist may have been from gram positive bacteria, although more study is required to verify the same. And so what we found out from this phylogenetic analysis of 221 tryptophanase orthologs is that gamma proteobacterial tryptophanase are well supported and having a distinct branch in the enterobacterials and vibrio branching that I am showing here, these three, right? Besides that, the presence of gram-negative bacteria and some of the gram-positive bacteria here with protist indicates that there had been horizontal gene transfer from prokaryote to eukaryote, and that led to an adaptation for the protist. And there is a study already done on dictyostelium, which is a protist, and people have identified two HGT cases in the protein that is categorized as beta LM lyase by the PFAM, and it belongs to an aromatic amino acid lyase. And tryptophanase is one such aromatic amino acid. So we believe that definitely at least dictyostelium, which is a protist, got tryptophanase gene from a prokaryote. And if you see the arrangements, you can see that the whole animal kingdom, which is represented here in this phylogenetic tree, you can see that there are three branches. So it starts with some first on the right hand side, you can see this blue color bars representing all bacterial system. Then we have red color fungi, animals, mold, plants, algae, as well as these protozoans and protists. Besides these, there are archaeal branch. So there is archaea, animals, and bacteria. And similar arrangement you can see here, where you have bacteria, different archaeal branch is different, and then animal branch is completely different. So it is quite coherent with the common classification that is based on RNA sequence. And when we are saying that protist got tryptophanase gene from horizontal gene transfer, we have to assign some function why a protist will take up another gene and get a load of it. So some of the HGT are classified as maintenance transfer. So the host gets this gene to get another additional metabolic advantage that will help them to survive. So for that, that type of transfer is called a maintenance transfer. But there could be innovative transfer where the host will get a unique ability to do some task. Okay, for example, in this case of tryptophanase, when a protist get tryptophanase from bacteria, as we are suggesting, so it helps the survival of the host, that is protist. And how it does is by utilizing pyruvate. We know that tryptophan degradation by tryptophanase will yield pyruvate, and pyruvate will directly go towards the conversion of towards TCA cycle and produce energy. So if protist have tryptophanase gene, the protist will degrade tryptophan, will get pyruvate, and will survive it in a competitive environment where suppose there is a lack of food. So it gives them additional biochemical activity to survive. Besides that, most of the microbial pathogens also possesses TNA. So this we already seen that Vibrio and other things, Pseudomonas and E. coli possesses TNA. And then we have the role of indoles in halophiles, and this aspect is, I think, is still untouched. Why halophiles have to keep tryptophanase in their system, because there is no role identified in archaea. So for protist I could identify the function, but for fungi and archaea the function is largely unknown. For fungus I can say that fungus produces various indole alkaloids, so fungus may utilize tryptophanase to produce indole alkaloids, that is the colored compound that fungus secrete. Okay, but for archaea the function is still unknown, so that would be an interesting study to carry out in future. So thank you so much everyone for listening. This was all about from my side about biofilm, the signaling that bacteria carry out and their advantages as well as some limitations, and some of the recent advancement that is going on in the area of inter and intra-species signaling. Thank you so much.
So, students, now you can discuss your queries quickly.
So good evening, sir.
Yes, good evening.
So is there any drugs that available in market which inhibits the quorum sensing signal, sir?
Okay, so no, I have never came across such a drug, because the only problem, as I mentioned, is it is just inhibiting the biofilm formation, but it is not killing the cells. Okay, so with quorum sensing, the advantage is that it will not kill the bacteria. So the logic behind developing quorum sensing inhibitors is that if you stop them communicating, but it will not, you will not inhibit their survival. You are not killing them, so there will be no pressure. So otherwise, what will happen? Bacteria will get pressurized that it has to survive, and due to that pressure, uh, it will generate some mutant. After some genetic makeover, it will develop a mutant strain that is not effective, means that will resist the antibiotic, right? But with quorum sensing inhibitors, what we are thinking is that we are just stopping their communication, but we are not stopping their cell division or protein synthesis or other vital biochemical processes. And since we are not able to stop them divide and stop them their survival, such drug is not in the market, because people uh, do not only want inhibition of biofilm, but people also want clearance of the pathogen from the body or from the site. So they want to kill the pathogen. Okay, so that's why the combinatorial treatment is the most said one, that a cocktail of antibiotics and quorum sensing inhibitors can be used for a better treatment of any pathogen. Say, for example, we have a quorum sensing inhibitor, we develop a composite of such inhibitors, uh, and then we can coat, so we can mix these types of inhibitor in a paint and coat the medical devices, medical implants in hospitals, and that will stop bacteria to form biofilm on such implants, or on the hospital area, wherever in the metal surface you find bacteria to grow. So you can coat those things, and it is a good aspect. But the industrial process is somewhat different. You need permission, you need FDA approval to use such things in a medical equipment, or as a drug, right? So as of now, to my knowledge, there is no such drug, but yes, people are developing various conjugates and derivatives of quorum sensing signals that can be used as QSI, but none of them, to my knowledge, has come to the market.
Thank you, sir.
Mhm.
Hello, sir.
Yes,
Yes.
Tejas.
Yes, sir. Sir, you said that nowadays these bacteria is also developing resistance and tolerance against these quorum sensing inhibitors. Then what is the advantage of quorum sensing inhibitors over antibiotics, sir?
So, like I said just now, that antibiotics target some vital process, whether it is a protein synthesis mechanism, or DNA replication, or RNA mRNA synthesis. So antibiotics target very vital processes, and that's why bacteria feel a pressure of survival, and that's why they develop resistance as well. Means only those types of variety will survive that cannot, means that are mutated enough to avoid these antibiotics. But with quorum sensing inhibitors, there is no such pressure of survival. We are just stopping them their communication, so it is less likely to develop resistance first. But still, as I showed you, that they are multilingual in nature, and since they are multilingual, what does that mean is they have multiple QS system. They can sense different types of signals. So even if we block one type of signal uh, communication, they can communicate with another system, and targeting multiple signaling system will become difficult. And this is just a chance. There is just one or two cases that has been reported, and that's why this area of QSI resistance is very recent area. You may have noticed that the article that we wrote is in 2014, and it was nearby that time 2012, I think. Research group reported such type of resistance, but it is not very well documented, and it is just an speculation that just like antibiotics, bacteria can also develop resistance against QSIs. But we need more and more data and study to have that. And as there is no QSI, means quorum sensing inhibitor, or the drug in the market, we don't know whether actual resistance happened or not. With antibiotics, we know that yes, there is actual resistance, as we have seen in Mumbai, in Delhi, like tuberculosis pathogen, and Mycobacterium pathogen, XDR strains are being isolated quite often, not only in India but across the globe. So by that we know, okay, it is means we are unable to treat that type of infection with all the sets of antibiotics that we have in our hand. Okay, but with QSI, we have never tested at such large scale, and so we don't know exactly whether this will happen or not. But there is speculation, and we have to take care of that while doing research on this aspect.
Okay, sir. Thank you, sir.
Hello, sir.
Yes.
Regarding the sir indole compound, sir. So, sir, indole compounds are mainly effective on E. coli bacteria, sir, or all type of the bacteria, sir.
Indole is a signal, and indole is a very unique signal. Tryptophanase gene is present in E. coli. It was first found in E. coli, and I think uh, it sets the baseline. So whenever we talk about tryptophanase, E. coli comes into picture because most of the strains of E. coli sequences is submitted to NCBI, and so this means that E. coli produces tryptophanase, and because it produces tryptophanase, it has the ability to degrade tryptophan into indole. So tryptophanase means more indole, right? E. coli secretes indole, and I have shown in my slide that there are many gram-negative bacteria. I think 50 bacterial genus are there that possesses tryptophanase. So this means they are able to secrete indole, right? And besides E. coli, there are Vibrio species that also possesses tryptophanase. But the interesting point with this is most of them were pathogens, and some of them were gut pathogens, and some eukaryotes that like blastocystis, that is also a protist, which is also found in animal gut, right? So mostly they were related to gut, and this also helped us to decipher or to at least link up why only gut pathogens have this ability. So the logic is animals cannot produce indole. Even as I said, plants also cannot produce indoles. So these bacteria actually serves as a source of indole for the host. And this is how this holobiont theory also exists. And if you will read about the holobiont theory, it says that humans or any animal for that matter, they evolve with bacteria because the presence of bacteria is everywhere. And you may imagine the type of spread. Just corona is just a virus, coronavirus, that COVID-19 that we are facing. Everybody is now know that it is spread a lot. And similarly, all the unicellular organisms also spread quite frequently. Whenever we are talking in our body, on our body, there are a lot of bacteria. Even whatever we do, we cannot get rid of them. And some of them are so symbiont that you can't remove them from your body. And gnotobiotic study is such aspect where you can try and study such things, right? So it is not the only E. coli, but there are other bacteria also that can produce indole. And gnotobiotic study is one such thing where you can distinguish whether it is a holobiont, means it belongs to a holobiont system or not. So to you all, I want to mention about the gnotobiotic study. What does that mean is, from the birth itself, you are keeping an organism in isolation so that it don't interact with outside bacteria or outside environment. So you give it all the things that it need, but sterile. Even the food will be sterile, so there will be no chance of any outside bacterial infection. So you have a facility in a gonotobiotic study. You have a facility where you are growing mice, and the moment it delivers a baby mice, you isolate it aseptically, so there is no infection, and then you keep it isolated in an isolated chamber so that no further bacterial or any type of microbial infection can happen to that organism. And then you study the effect of some drug treatment or any other thing, just to study whether gut bacteria have some role or not. So those kinds of study are unique, quite important, but hectic as well, and it is expensive too.
Okay, sir.
So
Okay,
I hope, Devraj, yeah, I answered your question that not only E. coli, but there are many other organisms
yes,
that
yes.
produces indole and communicate.
Or sir, indole sir are two type, no sir? Either synthetic or naturally, sir.
No, yes, synthetic indoles are there, and since we got to know about these signals, indoles are interspecies as well as inter-kingdom species signal. Means indole produced by E. coli can affect Pseudomonas can affect Burkholderia, Candida, many other organisms. So that's why people started developing indole derivatives, but they are not natural. Okay,
Yes,
and
yes.
what I was talking about is all naturally or biologically converted indole derivatives like indole acetic acid, indole propionic acid, etc. People are synthesizing indole compounds that are QSIs as well, and they are developing it so that we get a very effective compound that can inhibit the quorum sensing signal.
Also, sir, derivatives are from the neurotransmitter, like sir, serotonin.
Yes, yes, 5-HT, I talked about the 5-HT, and 5-HT can be synthesized in our brain directly and in the
Yes.
peripheral system as well. Okay, so what bacteria helps is bacteria helps in the level of peripheral 5-HT, not the brain, not the 5-HT that is available to brain. And 5-HT cannot transfer the blood-brain barrier. So that is also one limitation. So it is not that indole can directly help to increase the 5-HT level in the brain and control our emotion or uh, change our mood, things like that. So 5-HT, peripheral 5-HT is governed by the gut pathogen, gut microbes, not pathogens. While the 5-HT that we synthesize in our brain is by our cells itself, but that is not indole. That has the indole group, but uh, it doesn't mean that animal can synthesize indole. It is an indole compound, but 5-HT synthesis is dependent on tryptophan.
Okay, okay, sir. Okay,
Mhm.
thank you, sir.
Any more questions?
Hello,
Hello.
sir.
Okay. Yes,
Yes,
Tej.
Tejas.
Sir, this question is not regarding the topic. Just I'm asking out of curiosity. You said that you did your PhD in CSI or IGIB, but how did you went on to become a professor in University of Korea, sir?
I didn't get you.
Sir, you did your PhD in CSI or IGIB, but
Yes.
how do you went on to become professor in University of Korea, sir?
Okay, so in research we have to do postdoctoral, means post PhD we have to do research, and then after experience in some university also offers a tenure track position. So similar positions is I am having, and I am an assistant professor, not professor, and professor level is different, and with experience we gain that position. But I am an assistant professor on a tenure track basis. Uh, and yeah, based on the publication and citation plus their interest, you get such position. And even other countries also have such system of tenure track in Canada, US, Europe, everywhere. So post PhD, you need some experience. Even in India, in Indian academic system also, you need some post PhD experience to get recruited. So this is just an academic setup. There is nothing, and it is a streamlined whether you are from biology background or any other stream, science stream. This system holds true for everyone.
Okay, sir. Okay, sir.
Yeah.
Hello, sir. Sir, my last question, sir. Can I ask, sir?
Yes, sure, sure.
Sir, about the sir biofilms. Okay, sir. So many sir biofilms contains the protein, lipid, DNA, and polysaccharides, sir. Okay, sir. And then sir
Yes.
mainly more than sir 90% contents of the water, sir.
Yes.
Okay, okay, sir. So, sir, if I want to inhibit the sir biofilms and kill the biofilms, okay, sir. So I know the 97% contents of the water. Then if I completely dry of the biofilms, can it inhibit, sir?
Yes, you can dry the whole system out, but thing is the applicability where you want to apply such system. If it is a water body and For example, your tank in your house, there also biofilm forms, right? Maybe it is algae, but even bacteria will also be there in the tank, or any other, suppose membrane filters. Biofilm forms on membrane filters as well. So you cannot use such approach, as you said. The drying out approach may not be first economical at industrial setup. For example, your fermenter also have a lot of bacterial biofilm present in them. If you are running a lot of fermentation activity, there will be a thin film. If you are using membrane filters, there will be the membrane filters in our water purifier. They get choked by such biofilm, if not on the dirt, but bacterial biofilms also choke it. So if you are using such approach, it may also damage the membrane, right? So such approach is not feasible in all location, right? For example, even in human body, cystic fibrosis patient is a standard example. Pseudomonas causes cystic fibrosis. Pseudomonas is an opportunistic pathogen. So what it does in an immunocompromised patient, its infection increases, means it grows more in number, and then it also releases toxin, so that decays the uh, patient body and its response to the treatment, right? So in cystic fibrosis patient, pathogen infection of pseudomonas increases. Now how you will use such approach in a body? You cannot use it. So that's why people try to uh, put antibiotic plus some quorum sensing inhibitors or other type of molecule that will stop their communication or growth of bacteria, like that. So in a normal body, uh, means animal body, we cannot use such approach. Even in a monument, suppose you have in monuments, bacteria, these microbial biofilms are there, and they start corrosion. If it is suppose metal body, or even uh, if it is just rock, they start secreting things that will degrade the rock, right? So in such cases, how you can use heat, because if it is a precious monument, you cannot apply such a high heat just to dry out the things, right? And also remember that bacteria, if it is a gram positive bacteria, they form spores, and spores are tolerant to heat. And even if you you make them dead, but spores will survive, and later on they will again infect somewhere else. So this type of cycle continues, and it has been for years. Bacteria, microbes had been surviving for years, maybe million of years, and we appeared later. So you can imagine that we cannot get rid of them, whatever we try. And that's what I am saying is, even in gnotobiotic study, it has been found that we get some bacteria, some useful bacteria from our mother while giving birth, or even sometimes in very few studies they are also saying that through placenta also, minor quantity of bacteria, the first liquid that we gulp, we get some bacteria from the mother, right? So ultimately it is a kind of unavoidable situation, but the strategy that you propose is quite unrealistic.
Okay, sir. But sir, it may apply, sir, in on the sir surface area.
Yes,
Not
definitely on an open surface, definitely you can try this. And but the thing is, the microbes will not grow on an open surface. This type of biofilm will only happen in an humid area, because bacteria have to survive. They have to get nutrient, and in a liquid medium they thrive well, right? So in a humid environment they get nutrient from the atmosphere, so only in those places they grow. The moment you apply heat and on an open surface, yeah, you can use that, but it is very rare to get such encountering such biofilm on an open area.
yes, yes, sir. Okay, sir.
Mhm.
And sir, dental animal is also considered as a sir biofilms, no, sir?
Yes. Periodontal disease is comes under the biofilm, and the pathogens or the bacteria that inhabit our dental plaque is also quite robust. Uh, So rather, sometimes ago, I had a feeling to develop a toothpaste that will contain QSIs, so that we will not form dental plaque quite often. But later I realized that in 2016, I guess some research group already have made that type of a startup, and whether it is realistic or not, they may have studied more than me. So I gave up, because it is not only about the QSI but also the bacterial growth that we have to stop, right? Uh, you can stop the biofilm formation, strong biofilm, strong plaque formation. You can stop that, but still, if they are in the oral cavity, they will grow by the nutrient. So there should be some other method as well besides keeping QSI type of molecule in the toothpaste. But that is a good
Yes,
idea
sir.
as well. Yeah.
Yes, okay, sir. Okay. Thank you, sir. No more
So
questions, sir.
I think we'll take last question. So there is a question in chat box that silver nanoparticles is used to eradicate the biofilm from medical devices. Will it affect the human body as silver is used in various medical devices? As silver metal and silver nanoparticle both possess different properties.
Uh, yes, this had been quite this type of approach of nanoparticle, and the debate about the toxicity, the silver toxicity, had been on for a long time. But I don't have the exact answer for this. But all I can say regarding the biofilm is that silver nanoparticle, the penetration of nanoparticles within a biofilm is known, but it is not only silver nanoparticle, but chitosan nanoparticle and other biopolymeric nanoparticles are also being tested for their efficacy in stopping the biofilm or degrading the biofilm or dispersing the biofilms. So it is not only the silver nanoparticles. Yes, they are being used, but I don't know about its commercialization, because you need FDA approval, and this metal toxicity, whether it is silver or another metal toxicity is an issue. So unless FDA approves it, we can't use such things on directly on implant. There are some formulations that people quote. For example, I said about fatty acids. Fatty acids are natural biofilm dispersing agent. So if you coat medical devices with fatty acids, it may help, plus it may not cause anything because it is a natural molecule. So it won't induce any type of allergies or something in our human body, right? So such thing people try, and that's why synthetic chemicals, synthetic quorum sensing inhibitors, they have to go through all the steps of approvals. But if it is a natural chemical, we may get the approval quite fast. And whatever it is, silver nanoparticles are antibacterial, and they are rarely used as a antibiofilm agent. So because they are antibacterial, this means they will kill randomly. It is a type of broad spectrum antibacterial agent. So because bacteria itself is dying, so biofilm will get reduced. So that type of activity will happen, but not silver nanoparticle alone cannot degrade biofilm. Yeah, I hope I have answered question of Nilansu Gaurav.
Yes, sir. Thank you, sir.
Okay, so I guess there are no more questions. And if you have any query, you can drop down the message or you can email me. Thank you so much, Dr. Prasun, for your time. And once again, you have accepted our invitation for this workshop. And we always look forward for your contribution in